So today we're going to be talking about circular dichroism, which is a method for determining secondary structures for proteins. So looking at alpha helix or beta sheets that might appear in a structure. So real quick overview of what this looks like is we shoot polarized light at a protein sample, and then we're going to get a directionality shift for that polarized light, either to the left or the right. And what this tells us is what type of structure we're actually seeing in our protein. So to get this up and going, we're going to start up a nitrogen tank, and this is going to allow nitrogen gas to flow through the system. And then we're going to go ahead and turn on the lamp as well. And we need to stabilize the lamp, so make sure you do that. And then we're going to open up the system and just make sure that everything is good to go. Now we're going to use a cuvette to actually test our samples. So we're going to make sure to clean that out with water and acetone. And then we can load in 200 microliters of our buffer solution to this cuvette. And you can see we have to do this pretty carefully. We don't want any air to be trapped in there. And we're going to gently tap it down before loading it into the system. And now we're going to do a test run with just the buffer. And as you can see, it's scanning through the different wavelengths right now. And this is checking for the high voltage count, which we just need it to be below about 600 in order to have a good sample. Now we're going to do a test run with our buffer. So we're going to run this 3 times. As you can see, it's going through 3 different times and 3 different reads right now. And once the different reads are done, we're going to load them into the analysis section. And we're going to take the average of these 3 reads, and this is going to be used for our blank for our buffer solution. So we're going to go ahead and add that. And now we can test with our protein after that. But first we need to also make a standard for our reader. So we're going to put our cuvette into the reader and allow it to get a blank so that we know how much protein we have moving forward from there. Now we're going to just do this all over again with our protein and load in 200 microliters into the cuvette and then let it run and then put it into the carry again in the cuvette and see what our actual concentration of the protein is. So once we get that all done, we're going to be using this nifty program that Ulva Pearson developed, where we can take the raw data and put it through this program called bkmerge. So we're going to put all of our data into a directory with bkmerge in there, and with this very simple command, get all of our data done real quick. And then we can load it into the spreadsheet, again, that Ulva created, and it does a lot of the analysis steps for us. So we just put in the different values that we get from those spreadsheets, and what we'll be able to see is once we put in the concentration that we have, we are able to get a very clean graph for our data.